Enzymes. Enzymes are biocatalysts which enhances a biochemical reaction. Uh, Almost all enzymes are proteins, but there are some nucleic acids which also behave like enzymes, and these are called as ribozymes. An enzyme, we can see with the help of this diagram, and an enzyme is just like any protein which has a primary structure. You can see here in this diagram, this is an enzyme which is having a primary structure like that of a protein. And when you look at a tertiary structure, we'll notice that the backbone of the protein chain folds upon itself, and these chains crisscross itself, and this makes many crevices or pockets they are made, and one such pocket is the active site. So an active site of an enzyme is a crevice or a pocket into which the substrate fits, and thus enzyme through their active sites enzymes through their active sites, they catalyzes a biochemical reactions at a very high rate. So here you can see that the substrate, they can fit into the active sites, and then it gives rise to an enzyme substrate complex. So enzymes catalyzes a biochemical reaction, but it differs from an inorganic catalyst reaction. Now, how does it differ from an inorganic catalyst reaction? So enzymes, they catalyze. One major difference is that inorganic catalysts, they work efficiently at higher temperatures and high pressures, whereas enzymes, they get damaged at very high temperature and pressure, maybe say above 40 degrees Celsius. But enzymes, they are when they are isolated from the organisms which normally live under extreme high temperatures like hot vents, sulfur springs, then they are stable and they retain their catalytic power even at very high temperatures up to 80 to 90 degrees Celsius. So thermal stability is an important quality of such enzymes which are isolated from thermophilic organisms. Chemical reactions. Now, and how do we understand the enzymes? Let us understand a chemical reaction first. Now, chemical compounds, they undergo two types of changes: a physical change, which simply change the shape by breaking the bonds, and it's a physical process. Another physical process is a change in the state, like when ice melts into water, or water becomes a vapor. These are all physical processes. But when bonds are broken and new bonds are formed through transformations, then that is called as a chemical reaction. For example, barium hydroxide, it reacts with the sulfuric acid, gives rise to barium sulfate and water. This is an inorganic chemical reaction. And hydrolysis of starch into into glucose, that is an organic chemical reaction. So rate of a physical or chemical process, it is the amount of product formed per unit time. So it can be expressed as this. So rate of a biochemical reaction can be the amount of products formed per unit time. So the rate can also be called as the velocity, or the direction is specified. So rate of physical and chemical processes, they are influenced by temperature among other factors. So the rate doubles or decreases by half for every 10% change in direction. So catalyzed reactions, they proceed at a very high rate as compared to the uncatalyzed reaction. So when enzyme catalyzes a reaction, so the rate would be higher than the same, but than the uncatalyzed reaction. For example, here you can see carbon dioxide reacts with water under the effect of carbonic hydrase enzyme and gives rise to carbonic acid. So in the absence of this enzyme, this reaction is very slow, and without but by using the enzyme present in the cytoplasm, that is carbonic anhydrase, the reaction speeds up. It is about 600,000 molecules which are being formed every second. So enzymes, they accelerate the reaction rate by 10 million times, and the power of enzymes is incredible. So there are thousands of types of enzymes which catalyzes the metabolic reactions, and there are multiple steps chemical reactions, and each step is catalyzed by some enzyme complex or different enzymes, and that is called as a metabolic pathway. Like glucose is converted into pyruvic acid. This is actually a metabolic pathway in which glucose becomes pyruvic acid through 10 different steps, and all these steps are enzyme catalyzed reactions. So when we'll be studying respiration of chapter 14, then we will study all the reactions, how glucose is converted into pyruvic acid. How do enzymes bring about such high rates of chemical conversion? So we have to see that with substrates they change into products. Okay, so enzymes they have active sites, and these active sites, substrates, they get fixed up and get converted into products. So substrate has to diffuse the active sites, and therefore there is an enzyme substrate complex which is formed. So here E stands for enzymes, and S stands for the substrate, and P stands for the product. So we see that the structure of the substrate get transformed into a product, and the pathway of this transformation is called as the transition state. This is called as the transition state, and there are many more some states between the stable substrate and the product. So we find that all other intermediate structures, they are unstable. They are unstable, and stability is something which is related to the energy status of the molecule or the structure. Hence, when we look at this graph, we see that the y-axis, it represents the potential energy content, and the x-axis represents the progress of a reaction, which is actually the transition through the transition state. We notice here two things: the energy level difference between substrates and products. This energy level difference between the substrate and the products, and whether it is exothermic or spontaneous reaction, or an endothermic or a repairing reaction, the S has to go through a much higher energy. So it has to go through a higher energy state or transition state. So the difference in the ever energy content of the S from that of this transition is called as the activation energy. So enzymes, they bring down this energy barrier and making the substrate to become products, and this process becomes easy. Nature of enzyme action. Each enzyme has a substrate binding site to its molecule so that it can become enzyme substrate complex, and it can change into product. The substrate, it has to bind to the active site of the enzyme fitting into the active site. So binding of the substrate induces the enzyme to change its shape, and it tightly bonds to the substrate. So the active site of the enzyme and the substrate, it breaks the chemical bond, and the substrate becomes the new enzyme product complex that is formed. So the enzyme releases the product of the reactions and free the product, and the substrate, they run through this catabolic cycle once again. There are various factors that affect the enzyme activity. These are temperature and pH, concentration of substrate, inhibition, and we'll talk about competitors. So temperature. Each enzyme show a high activity at a particular temperature and pH, that is called as the optimum temperature. And low temperature preserves the enzyme in a temporarily inactive state, whereas the high temperature destroys the enzymatic activity because proteins are denatured by heat. Okay, then when we see the concentration of the substrate, we find that the reaction reaches a maximum level if there is increase in the substrate concentration. So the velocity or the speed of the reaction of the enzyme, it increases, but it is not exceeded any further with the rise in the concentration of the substrate. This is because the enzyme molecules, they are fewer than the substrate molecules, and after saturation of these molecules, there is no free enzyme molecules to bind with the additional substrate molecules. So the activity of enzyme is also sensitive to the presence of specific chemicals that binds to the enzyme. When the binding of the chemical shuts off the enzyme activity, that process is called as inhibition, and that chemical which inhibits the activity of the enzyme is called as inhibitor. When the inhibitor resembles the substrate, in its molecular structure. So it inhibits the activity of the enzyme, and it is called as the competitive inhibitor. So due to its close structural similarity with the substrate, the inhibitor competes with the substrate. So the inhibitor competes with the substrate to bind for the binding site of the enzyme, and the substrate cannot bind, and so the enzyme action is declined. So inhibition of succinic dehydrogenase by malonate, which closely resembles the substrate succinate in structure. So such competitive inhibitors, they are often used in the control of bacterial pathogens. Classification and nomenclature of enzymes. Enzymes are of various types like oxidoreductases or dehydrogenases. They catalyze oxidoreduction reactions between the substrate. So one substrate is reduced and one is oxidized. Another is transferases. These are the enzymes which catalyzes the transfer of a group between a pair of substrates. Hydrolases are the enzymes which catalyze the hydrolysis of ester, ether, peptide, glycosidic or different bonds. Lyases, enzymes that catalyzes or removal of groups of substrate, then they are called as hydrolases. Isomerases, they catalyze interconversion of optical, geometric or positional isomers. Ligases, they link together to two compounds. Cofactors. We know that enzymes, they are made of polypeptide chains, but a number of cases are there where the non-protein constituents, they are also called as cofactors, they bound to the enzymes. So three kinds of cofactors they are identified. These can be prosthetic groups, coenzymes and metal ions. Prosthetic groups वो होते हैं जो organic compounds हैं. They are distinguished from the other cofactors because they tightly bound to the apoenzyme. For example, peroxidase and catalases. They catalyze the breakdown of hydrogen peroxide to water and oxygen. Coenzymes, these are the organic compounds, but their association with the apoenzyme is only up to a certain level, and coenzymes, they act as the cofactors in a number of different enzyme catalyzed reactions like NAD, NADP, nicotinamide, adenine dinucleotide phosphate. A number of enzymes, they require metal ions for their activities, which form the coordination bonds with the side chains of the active sites, and they also form more coordination in bonds with the substrate. For example, zinc is a cofactor for the proteolytic enzyme carboxypeptidase. So catalytic activity is lost when the cofactor is removed from the enzyme, which says that it plays a crucial role in the catalytic activity of the enzyme.